Okay, before we begin, are there any questions? Not hearing anything, so let me share my screen and then we'll begin. Note, I sent out an announcement telling you to, or advising you to finish the unknown notes pages. Some of you don't have your last results up, and this assignment will be graded. It's 5 points. It's like a normal lab. You should be seeing my web page now. Today is March 7th, and we're getting near the end of the term. We're supposed to be finishing chapter 13. I don't think we'll succeed in doing that, but we'll see what we get. If we don't finish chapter 13 today, and we especially have a large number of slides left, then I'm going to move this review over to the lab, and we'll give the review in the lab time. Okay? If we finish chapter 13 or get close to it, I won't do that. So just to let you know what's going on. The unknown notes pages closes this Friday, March 8th. The unknown project, the final report is due at 11:59 p.m. this Saturday. Any questions about any of that? All right, let's go to the lecture. So I talked about the T4 virus having this shape. It's the virus that's been studied the most, more than any other virus. And so we'll talk a little bit about viruses by using what's known about T4. When a virus infects an animal, this will stimulate the animal host to produce antibodies. Antibodies recognize and bind to viral surface proteins that we call antigens, and these will be the outermost molecules of the virus or whatever the antigen is. And I guess not antigen, but whatever the object is that the host immune system is recognizing and attacking. So antibodies will recognize and bind to the viral surface proteins and glycoproteins. Some viral proteins can change or mutate frequently, and then different viral strains can infect the same individual more than once. An example is the flu, which it mutates so that every something like third year after you get the flu shot will no longer protect you from the strains of the flu that are going out. So they recommend you get the flu shot every year, but you could go like every 2 years or something like that. Because on the third year, the flu shot really will not protect you much. And of course, we're giving an oversimplification here. Let me see if I can diagram what happens. When you first get a flu shot, and this will be the time on the x-axis, maybe I can write that, sort of. A dot there, time. And then on the x-axis is the percentage. I'm not going to try and write that one. Percent immunity, with 100% being, of course, total immunity. You never get 100%, but you get it high when you first get the flu shot or whatnot. And then your immunity drops with time. Oh, come on. Go back home, switch that. There we go. So your immunity drops with time. And in the flu shot, the immunity after 3 years drops below a threshold number that they say the flu shot's not giving you any protection anymore. And I don't know what this number is, if it's 37% or 20%. But below that number, they say the flu shot is not giving you protection anymore. In reality, it's giving a little bit of protection for a little bit longer, but at 3 years, they say the immunity has dropped. And like I said, it never reaches 100%, even in very young adults or teenagers. They're probably the best population to get immunity. You never get 100% immunity, but it's usually very high among young adults and teenagers, reaching over 90%, probably about 95%. Any question about that? As you know, COVID-19 changes too, and that's why we will probably need to get a COVID-19 shot about every year if you want to be immunized against COVID-19. So that's about the same as the flu. The flu actually mutates a little more rapidly than COVID-19. Other viruses do not tend to mutate very rapidly at all. For example, we're still using the vaccines made against polio and what else? We'll just say polio. I'm drawing a blank here for another vaccine that was made in the 1950s, both of them, and they're still being used today because they're still effective, meaning this virus has not mutated. How many years is that? 50 years plus about 20 years. So that virus has not mutated significantly in 70 years. We're still using the vaccines today and they're still effective. Another one would be yellow fever. That vaccine was made at least in the 1950s, if not the 1940s, and it's still good today. Any question about any of that? And it's just some viruses mutate more rapidly than other viruses. Unfortunately, we're not going to talk about the flu vaccine, but it has a reason for why it mutates more rapidly than most other viruses. When we're talking about viruses, it's important to talk about viral taxonomy. Viruses are grouped into families based on the type of nucleic acid, the replication strategy, and their morphology. Viruses, let me see if I can remember this. King Peter's son came over from. So viruses are grouped into, I think it's class, just above, or maybe it's order, just above family. But higher levels of viruses, we do not classify them, with the exception of what domain they infect, meaning we do classify viruses, animal viruses or plant viruses. So the domain, not the domain, the kingdom they infect, sorry, the kingdom they infect. We do classify viruses by the kingdom they infect, but we do not know the evolutionary history of viruses, and their genome is not very long. Plus, viruses quickly evolve with their host species they infect. And that means viruses, we just don't have a very long evolutionary history of viruses, and we don't know where some viruses came from. And so we can't classify them into higher orders like, well, subkingdom or phyla. We just can't because we don't know their evolutionary history. Okay? They don't have enough of a genome that is not mutating. And so we can't say where the viruses came from, what they were, like, I don't know, tens of thousands of years ago, or maybe hundreds of thousands of years ago. We do classify viruses into families. If they have a family name, the name is viridae, like herpes viridae for the herpes family of viruses. The genus name of a virus is virus. So for a herpes virus, it's actually genus name, herpes virus. Then there's viral species. These are a group of viruses sharing the same genetic information and ecological niche. An example would be the polio virus, infecting humans. That's its ecological niche, who it infects. And of course, they're fairly genetically similar to each other, all the members of the species. And viruses are only known by their common English names. They have no Latinized names. And wherever you are in the world, regardless of what language you speak, when you're talking about the polio virus, it is called poliovirus. Why is because apparently the first people who studied virus were all English speakers, and then they started giving viral names, and they called them English names, common English names, like poliovirus. And then when other people started studying viruses from other languages, they just used the names that had already been given to the virus. So the French call it the polio virus. Any question about that? And there are no Latin names. Now, viruses do have subspecies. They are designated by a number, like there's poliovirus type 1, poliovirus type 2, and you guys have probably never heard of those. But I know you've heard of herpes virus type 1, herpes simplex virus type 1 and herpes simplex virus type 2. They are the same species of virus, meaning they share the same genetic information and ecological niche. They do infect human cells. It's just that they are a little different. One is usually in, oral or mucous membrane infective virus. The other is usually a venereal disease, which is a mucous membrane, by the way. And herpes simplex type 1 can hit the genitals, the mucous membranes of the genitals, and herpes virus type 2 can hit the mucous membranes of the nose and the mouth. Any questions about any of that? All right, so how different are different viral subspecies? With poliovirus type 1, when it infects people, it causes the disease polio. When poliovirus type 2 infects people, it vaccinates them against the poliovirus, meaning the live vaccine, which has a live virus in it, that we use as a vaccine to prevent people from getting polio, is poliovirus subspecies type 2. And that tells you how different the subspecies can be. One subspecies causes the disease polio, the other subspecies we use as a vaccine to prevent getting the polio virus. Any question about that? And that subspecies is, if I remember correctly, it is the Sabin vaccine for poliovirus. All right, so an example of viral taxonomy, the family is herpes viridae. The genus would be the herpes simplex virus. The common name, which has not been used by clinicians, the general public, and even scientists are stopping to use the common name. The common name was human herpes virus, which is more often called a herpes simplex virus. Any question about that? And the subspecies is herpes simplex virus type 1. So there's an example of viral taxonomy. Any questions on viral taxonomy? All right, this is something that I've done, growing bacteriophages in the lab. Remember, a bacteriophage is a virus that infects bacteria, and how you can grow them in the lab is you can plate out a plate with a lawn of bacteria, and that's why this plate in this region here, let me blow this up. In this region here, it looks white because that's from the bacteria growing on the plate. And then you spread over the plate after you put the bacteria on it. You haven't grown the bacteria yet. You just plated them. But before you incubate it, you then put on a solution containing a virus and then spread that around. Wherever the virus lands on a bacteria cell, it will then infect that cell and replicate it, burst that cell, and where will those viruses go? Neighboring bacteria cells, which will then infect them, replicate, burst those cells, and you can grow that up on a plate, and all of these plaques are where a virus infected a bacteria cell and then grew to form a plaque, lysing the bacteria cells. So wherever we see a plaque, there are no bacteria cells. Around the plaque, there are bacteria cells. Any question about any of that? And from this, you can then determine what the virus concentration was in your original solution. You may have made a dilution before you plated it, but if not, then that's the actual number of viruses. And this would be like, about 300 viruses per mil on this plate here. And you see how some of the viruses are different. That one, if you look at that closely, let me blow that up a little more. This one looks like there's 3 plaques that grew to form one, but that one's a very large one. That's a very large one. And then where's a small one? There's a small one. What might be different on the sizes of the bacteria is this plaque got started with a virus infecting the bacteria immediately. And so it went on to make a large plaque. This virus did not infect the bacteria immediately. What happened was, let's make this our surface, and that is there was a bacteria cell over here, and then one over here, or further over, and a virus went near that cell, but not at the cell. But then this cell starts growing, and so it'll grow a cell here, grow a cell here, grow a cell here, grow a cell there, still not touching the virus. But then the bacteria grows further, and now it touches the virus, and the plaque would then start. The viral replication would occur, the cell would be lysed, and then the viruses will go out and infect other cells, causing the start of the plaque to form. So this plaque may have started later than that plaque, and that could be a reason why that's a smaller plaque than this one. Any question about any of that? So that's how you can grow up viruses in the lab, at least bacteriophages, and that is you have them infect the strain of bacteria that they're supposed to grow in, what their host cell is, grow it up, and then you can get different viruses. You can cut out this portion of the agar and purify the virus, or you could just swab this portion and then put it, I don't know, wherever you want to put the virus, on like on bacteria cells or what. All right, let's talk about viral multiplication. The viral genome only contains a few genes, including the capsid proteins necessary to make the virus, and a few other genes that may be needed for new virion synthesis. What the virus does is it infects a cell and then hijacks the cell, forcing the cell to make new viral copies. Following infection, viruses take over the host cell's metabolic machinery, use the host cell's ribosomes, tRNA, amino acids, ATP, and some enzymes to replicate the virus. The basic mechanism for viral multiplication is similar in all viruses. For bacteria viruses that we call bacteriophages, they follow two life cycles where the viruses infect a cell and then replicate in the cell using one of these two mechanisms. There's the lytic life cycle where the host cell is infected and then lysed, it's killed and then lysed, releasing the newly replicated viruses. And then there's the lysogenic life cycle where the virus replicates, but it does not kill the host cell. In the lysogenic life cycle, as long as that life cycle continues, the virus and its host cell live in symbiosis. So the host cell remains alive. Any question about any of that? All right, let's talk about the lytic life cycle first. To make new bacteriophages, what do we need? What components of the virus need to be made? Can anyone answer that? Don't have time to wait. So you need to replicate the viral genome. For simplicity, I'm only going to show you, what is it? One, two, I think there's 4 RNA. So only one of the 6 possible viral genomes, double-stranded DNA. You replicate the double-stranded viral DNA, and you've got newly replicated viral DNA, the genome. You have to transcribe the viral DNA to make the viral messenger RNA, to make the viral proteins, including the capsid proteins. The capsid proteins then need to come together with the viral genome to make the new progeny virions. So that's what you need to do to replicate a new virus or new virions. Any question about any of that? All right. The viral multiplication cycle in the lytic life cycle follows 5 stages. First, there's attachment, where the phage actually attaches to the host cell. Second, there's penetration, where the virus penetrates the host cell and then infects the host cell. We'll talk a little bit about that. I'll talk later about it. Then there's biosynthesis, where the virus actively replicates its genome and the viral proteins. Then there's maturation. This is the assembly of the new bacteriophage particles. And then there's release. In the lytic life cycle, the virus encodes the enzyme lysozyme, which breaks down the cell wall. That will eventually cause the host cell to burst, and that will release the newly replicated viruses, the virions. Any question about any of that? If not, let's watch a little video discussing the lytic life cycle.
First, the phage particles stick to the surface.
And unfortunately, in the movie here in this player does not show. So let me go to another. I think I've got another movie.
First, the phage particles stick to the surface of the bacterial cell.
Oh, sorry, I hit the wrong one.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
All right, let's stop here and we'll continue with this movie for the lysogenic life cycle, but that's the lytic life cycle. Any questions about any of that? There's a lot going on in this little movie, so let me come back here and I'll come and show you a few parts of it. So first, the virus has to attach to the host cell. And as I mentioned earlier, not today, but 2 days ago, you have specific molecules that are called ligands or adhesions on the virus that bind to specific molecules on the host cell called receptors. And in this virus, the adhesions for the virus will be on the tail fiber. Let me blow this up. So the adhesions will be right here and right there and right there and right there. So they're on the bottom part of the tail fibers for this virus. And the receptors are on the host cell, and they will be underneath the tail fibers. So there will be a molecule here, a molecule here, a molecule here, and a molecule there. So that's how the virus attaches to the host cell. And if the host cell lacks those receptors, the host cell cannot be infected. In the case of HIV, it's actually binding to receptors that are markers of the helper T cell. So that's attachment. Then there's penetration. And you saw that the virus gets rid of the cell wall under this region. It doesn't actually rotate the way that little movie showed. What it does is it secretes a little bit of the enzyme lysozyme right there, and that digests the cell wall. So this region right here will not have, come on, mouse. Sorry, my mouse died. This region right there will not have a cell wall because of the lysozyme. And then the sheath of the virus contracts, which is trying to show you right here. This is the, come on, mouse. This is the virus when it's not contracted. This is the virus when it's contracted. That puts pressure on the head here, and that will actually inject the nucleic acid or the genome of the virus into the cell, into the cytoplasm. And we actually saw that. Let me end this and come back to the movie. I'm not seeing the movie now. Maybe it's here. Nope. There it is. For some reason, I couldn't see that. Oh, shoot, I got to play it to see it.
Type of bacteria. Next, the phage breaks down the cell wall.
All right, so that's
And the nucleic acid from this
right there.
capsule is type of bacteria. Next, the phage breaks down the
So that's by the enzyme lysozyme breaking down the cell wall there.
cell wall.
And
And the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The new phages release an enzyme which weakens the cell wall, and the host cell bursts. This process of viral multiplication is called the lytic cycle.
There we go. All right, let's continue. Oops, right here, I guess. So once the genome is inside the virus, the genome, usually what it does, particularly if it's a DNA virus and following the lytic life cycle, is one of the first genes expressed on the genome is to make an enzyme which digests the chromosome of the host cell. The reason is the virus wants to call the shots in the host cell. And if you digest the chromosome of the host cell, then only your DNA is calling the shots in the cell. And the virus, what it does is it replicates itself, meaning replicating the viral genome, and then transcribing that viral genome to make viral proteins. And that's this stage called biosynthesis. So the phage makes the viral protein molecules, especially the protein of the outer coat, any other proteins that are needed by the virus, as well as replicating the DNA. And then it stuffs the DNA into the head of the virus, and the virus's particles come together, they self-assemble to make the new virions. And then the virus has an enzyme which stuffs the viral DNA into the newly replicated virion. And it's that enzyme that stuffs the DNA which occasionally makes a mistake, and it may put a stretch of the host chromosome DNA into a virus instead of the viral DNA. And then the virus, what it does is the last thing it does is it codes an enzyme for lysozyme, which dissolves the cell wall, and then water will come into the cell, causing the cell to burst, because the cell membrane no longer has the cell wall to protect it. And then that will release the newly replicated virions. There's two terms you need to know for the lytic life cycle. That's the burst time. That is the time from initial infection of the virus into the host cell until the time when the cell will burst. And that will depend on the species of virus and the temperature mainly. But an average, the burst time is 20 to 40 minutes, which I would assume would be room temperature, but I'm not positive what the average temperature would be. And then the burst size is the number of newly replicated virions. This number will depend on the virus infecting the host cell, usually 50 to 200 newly replicated viruses burst out of the cell. Any question about any of that? All right, so that's the virolytic life cycle. Upon attachment of a bacteriophage to a virus, if the virus follows the lysogenic life cycle, then one of two things can happen. the lysogenic life cycle can enter the lytic life cycle. And that just follows the same lytic life cycle I just told you, where the virus infects the cell, uses it to make newly replicated viruses. The virus encodes for the destruction of the cell wall that lyses the bacteria, releasing the newly replicated virions. So the lysogenic virus can follow the lytic life cycle. Any question about that? The other thing that can happen is the lysogenic virus can continue the lysogenic life cycle by having the DNA of the virus integrate into the host cell's DNA, starting the lysogenic life cycle. While the lysogenic life cycle continues, the bacteriophage will remain inactive, latent. It will not enter the lytic life cycle. And how that happens is there's a repressor gene that shuts down the lytic life cycle and therefore continues the lysogenic life cycle. The bacteria host cell is termed a lysogenic cell, and the two remain alive with the virus inside the host cell's DNA for as long as the lysogenic life cycle continues. Any question about that? All right, let's continue watching the movie on the lysogenic life cycle. There it is.
Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates, and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, any questions about that? So initially, when a lysogenic virus, bacteriophage virus, infects a bacteria cell, it has to decide immediately, does it enter the lytic life cycle or does it enter the lysogenic life cycle? If it enters the lytic life cycle, it follows the lytic life cycle I just discussed a little earlier. If it enters the lysogenic life cycle, the DNA of the virus actually integrates into the host chromosome. This we call a prophage, where the viral genome is a part of the host cell's DNA, the host cell's chromosome. And then as long as that continues, where the repressor gene shuts down the lytic life cycle, the lysogenic life cycle will continue. And the only way the virus gets replicated is when the host cell replicates its DNA. When the host cell goes through replication, it'll make two molecules of DNA or two chromosomes. One will move to one side of the cell, the other to the other side of the cell, and then the cell will split down the middle. Both cells will get a copy of the lysogenic genome. And then this can happen for many generations, where the cell can stay in the lysogenic life cycle, where the host cell and the virus are living in symbiosis, living together. Any questions about that? All right. And then, as it was mentioned in the video, for reasons that are not fully understood, even after, I would say, over 70 years of studying this, we still don't have a good reason or a good handle on why the lysogenic life cycle stops. And then the prophage excises itself from the host chromosome, and then we have a separate molecule of the viral DNA as opposed from the host chromosome DNA. And then the cell or the virus enters the lytic life cycle. It will then replicate viral genome, replicate viral proteins, assemble them, make new virions, and then lyse the cell, releasing the newly replicated viruses. Some of them will then go on and infect a host cell, and immediately when that happens, the lysogenic virus has to determine, do I enter the lysogenic life cycle or do I enter the lytic life cycle? Any question about any of that? That's the two life cycles of a bacteriophage, a virus that infects bacteria. We already talked about prophage, already talked about the repressor proteins. And already talked about that slide, so let's move on. There are some consequences of lysogeny. First, a lysogenic bacteria cell, meaning one that has a lysogenic virus in it, is immune to infection by the same viral species that is currently inside it as a lysogenic virus. Any question about that? So once this cell has a lysogenic virus in it, it will be immune to infection to other members of the viral species. And then the second consequence is that the bacteria cell can convert its phenotype, meaning change its phenotype. The reason being is that this is the DNA of the original host cell, that is the DNA of the virus. This cell gets new genes, and they're the viral genes, and then that viral gene incorporates into the host cell's chromosome. When those viral genes are expressed, this cell can change its phenotype. And that's called phage conversion, where the host cell gains new properties once it is infected by a lysogenic virus. The best example of phage conversion is when Corynebacterium diphtherium produces the diphtheria toxin. This can only happen in the bacteria, Corynebacterium diphtherium, when the bacteria has a lysogenic virus in its chromosome. Because the Corynebacterium diphtherium toxin is produced by the lysogenic virus, meaning that's the gene making that toxin. Corynebacterium diphtherium, the bacteria that lacks the lysogenic virus, cannot produce the Corynebacterium diphtherium toxin. Any question about any of that? Okay, enough of talking about bacteriophages, viruses that infect bacteria cells. Let's talk about viruses infecting animal cells. And these are called animal viruses. Remember, each kingdom has different viruses that infect it. And we do classify viruses by the kingdom they infect. The multiplication of animal viruses surprisingly follows the basic pattern of multiplication of bacteriophages, but there are a few differences. For example, attachment is different. Viruses that are animal viruses only have to attach to the cell membrane. They don't need to attach to the host cell, degrade a cell wall, and then inject their genome into the host cell, because it's much easier infecting an animal cell because all they have is a cell membrane. So the penetration is a little different. We'll talk a little bit about that. Penetration by animal viruses happens by endocytosis or fusion. The encoding is pretty much the same. Biosynthesis is also of the virus is pretty much the same. Maturation can be a little different, especially if the virus multiplies in the nucleus. You'll have to get assemble with the viral capsids in the cytoplasm. So in a eukaryote cell, which would be animals, the virus can replicate in the cytoplasm or it can replicate in the nucleus. And like I said, assembly will be different if the virus is replicating in the nucleus, because the genome is replicating in the nucleus and the protein portion of the virus is being made in the cytoplasm. And then release can be different. It can be the same, meaning animal viruses can release the new virions by rupturing the cell. And these we call lytic animal viruses. So we do have the lytic life cycle in animal viruses. But the release could be different. And that is the, in animal cells, we have the budding life cycle. And this is a life cycle of virus we do not have in bacteria cells. The budding life cycle virus, they will all be enveloped. And we'll talk about why that's the case in a little bit. All right, so the main difference in the replication of animal viruses from bacteria viruses is that animal viruses may replicate in the nucleus or in the cytoplasm. And of course, bacteria viruses only replicate in the cytoplasm because that's the only region of the cell in a prokaryote cell. Any question about that? All right. So as a rule of thumb, if the animal virus is a DNA virus, it will replicate in the nucleus. Why? Because the nucleus has all of the proteins and things the virus needs to replicate its genome. And it's a DNA virus, so it's going to replicate it in the nucleus. And that's a rule of thumb. A DNA virus replicates in the nucleus. If the virus is an animal virus, I got to qualify that. If it's an animal virus, it replicates in the nucleus. If it's a DNA animal virus. If it's an RNA animal virus, the rule of thumb is the virus replicates in the cytoplasm. Why? Well, you need to replicate the protein, and the protein machinery is in the cytoplasm, so we may as well have viral replication in the cytoplasm. That's a rule of thumb, but there is one major exception that you must know, and that is the retroviruses are an RNA virus, but they do not replicate in the cytoplasm. They replicate in the nucleus. And we'll talk why later on, but probably not today, because I doubt I'll get that far in this lesson. All right, attachment of animal viruses has the virus attached to the cell membrane. How the virus can get into the cell or infect the cell is either by endocytosis or by fusion. Now, in earlier versions of the textbook, they didn't say that the virus entered the cell by endocytosis. They said by pinocytosis, and that was actually a mistake. The virus, an animal virus, enters the whose cell it infects either by endocytosis or fusion. Endocytosis is where the virus binds to the host cell's membrane, and then the host cell's membrane invaginates further, and then this will come together and reseal, reforming the cell membrane right here, and then sealing off this newly formed vesicle here, meaning the membrane will come together here, and that'll make a vesicle, bringing in the virus into the cell by endocytosis. The second way that the virus can enter the cell, I don't think I have a picture of that. Not really. So let me show it with paint. Oops, didn't get it. Let's go here. You get a virus binding to the cell, and the virus has a double membrane called an envelope, and the cell has a double membrane called the cell membrane. The two can fuse. When they fuse, let me see if I can do this right. They'll fuse, and then the cell membrane will come here. Oops, almost didn't do that right. And here. Let me make that a little bigger down here. Oh, that's actually connected. That's good. And now connect this one. They can then fuse. And as you can see, once they fused, make that a little connected better. The inside of the virus is now connected to the cytoplasm, so the two can come together. What's important is the virus here can move into the cytoplasm after fusion. Any questions about fusion or endocytosis? Not quite there yet. There we go. Another picture. Penetration. And this looks to me like this is endocytosis. This is invaginating further, and then it pinching off right there. So the cell membrane reforms, and that makes the vesicle, which moves into the cell. The capsid needs to be released, and the capsid has to be removed for the virus to then start replicating. But there are means to do that. All right, let's talk about another difference that animal viruses can have. An animal virus can replicate using the lytic life cycle, where the virus will infect a cell. The virus will then multiply inside the cell. And then the virus will code for the destruction of the cell, lysing it, which will release the newly replicated virions. So animal viruses can follow the lytic life cycle if they're a lytic life cycle virus. The other main life cycle that animal viruses have, which is absent from a bacteria virus, is the budding life cycle. In the budding life cycle, the virus replicates but not at large numbers, usually. Usually the virus replicates at low numbers, so the virus replicates slowly. And here we have a virus that replicated, and the viral genome is someplace over here, and it made a new copy of the virus, the viral genome, which got together with the viral capsid. The viral capsid then comes up to a membrane. And that region of the membrane it comes up to will be a membrane that has viral proteins and glycoproteins in it. And then the virus will begin pushing on that envelope, not the envelope, the pushing on the membrane. And that membrane could be the cell membrane, it could be the nuclear membrane, or it could be the membrane of the endoplasmic reticulum. The virus continues pushing, making a bud, which is actually seen here, the buds of the virus. There's a cell there, and it's got 1, 2, 3, 4, 5, 6, 7, at least 7 viruses budding out of it. And the virus can rip off a portion of the membrane, which could be the cell membrane, the nuclear membrane, or the membrane of the endoplasmic reticulum. And that is how the virus obtains its envelope, by ripping off a portion of the membrane from the host cell. Inside that membrane of the host cell, like I said, there will be viral proteins and glycoproteins, and the virus just knows this is the region to go to rip off part of the membrane with the viral proteins and glycoproteins. And then that's how the virus buds from the cell. That then will break off and then is free to, I don't know, circulate and then infect another cell. That's the budding life cycle. In the budding life cycle, the host cell usually has no problem with the virus budding off a portion of its membrane. The cell can usually lose a little bit of its membrane, and so the budding virus does not usually kill its host cell. So a budding life cycle virus of animal cells or animal viruses usually does not kill the host cell. If the host cell is killed, it's usually killed by the immune system, recognizing that this cell is infected and then mounting an attack against that cell to kill it. And how that happens is probably from the viral proteins and glycoproteins in the membrane of the cell membrane, at least, of the cell. The host immune system can recognize these molecules as being foreign. And if it does recognize it as being foreign, it can then mute an attack on this cell. Any questions about any of that? All right, that's the budding life cycle. And we also talked about the lytic life cycle. The lytic life cycle of viruses, of animal viruses, as well as bacteria viruses, are non-enveloped. And remember, we call those naked viruses. All of the viruses, the animal viruses that follow the budding life cycle, all of them have a envelope, because they obtain the envelope when the virus buds from the cell. Any question about any of that? Now, I do need to briefly mention that in animal cells, we do have other life cycles. One that is very similar to the budding life cycle is usually called the persistent infection life cycle. It is a naked virus, but this virus leaves the cell in a way similar to budding. What happens is the virus replicates in the cell, and then the virus gets incorporated into a cell vesicle, and then the cell vesicle will fuse to the cell membrane and then open up so the contents of the vesicle can then be dumped out of the cell. So this life cycle is very similar to the budding. It's just that budding doesn't happen. We have a vesicle which brings the virus to the cell membrane. The vesicle membrane fuses to the cell membrane, and then that will dump the contents in the vesicle outside the cell, which dumps the virus outside the cell. Any question about that? All right, let's move on. Oh, let me mention that the fourth life cycle is the latent life cycle. And the latent life cycle is when the virus is no longer metabolically active, does not cause disease. The virus goes into a state of hibernation and then just waits to come out again later. And then if it does, it'll cause disease. The best example would be either chickenpox going latent, and that can be for decades, and then the virus activates, becomes becomes metabolically active and replicates, and that causes the shingles. All right, viruses can have different genomes. We can have a virus with double-strand DNA, a virus genome with single-strand DNA, a virus genome with double-strand RNA, a viral genome with single-strand RNA. And remember, if we have a viral with single-strand RNA, you need to state whether it is a single-strand positive sense RNA or a single-strand negative sense RNA molecule. Each virus needs to have a slightly different mechanism for multiplication and biosynthesis depending on what its genome is. For a DNA virus that's double-stranded, it's very simple to replicate. You just follow normal DNA replication, meaning these double-strand DNA will separate into single-strand DNA. Each single strand will be used as a template to make newly replicated virion DNA. Any question about any of that? Obviously, it's a little different, and we'll talk about how it'll differ with the different, mainly it differs for the single-strand RNA, but we'll talk about all the others too. All right, let's talk about the first DNA viruses, and there's a classification of animal viruses, breaking them down into either DNA animal viruses or RNA animal viruses. This will depend on what the genome of the virus is made of. So there are DNA animal viruses and RNA animal viruses, and they have their respective genome for their cell, meaning a DNA animal virus has a DNA genome for the virus. There are 7 families of DNA viruses that cause disease in humans. This table, let me see if I can blow this up, is showing you the 7 families. You don't really need to know the table. We will talk just a little bit about the pox viridae. Herpes viridae I've already talked about a little bit. We won't talk about this one, that one. Adenoviridae we will talk just a little bit about, and that's because this family of viruses is the 3rd family of viruses that cause the human colds. Adenoviridae cause the human colds. However, they are the third family that causes the human colds, meaning this one has the least species in it that causes the human colds. The number one species causes the most colds, and then the number two causes the second most colds. And this is the number three, the third most common cause of the human cold. I don't think we'll talk about adenoviridae or herpes viridae. It's telling you the DNA type, double-stranded DNA or single-stranded DNA. This one's a little different. We won't talk about it. Whether the virus is enveloped or naked. The capsid symmetry, we're not going to talk about that. The size, we're not going to talk about that. And then the representative genera, meaning genuses, and the diseases that they cause. So with the Poxviridae family, there's a species in this family that causes smallpox. Fortunately, that virus has been eradicated in the world, so it only exists now in 2 labs in the world, as well as maybe in the Center for Disease Control, in which case they're not even studying it, it's just in the lab. And it's also in the US Army lab, Biological Army lab, and then the Russian Army lab. The only places where the smallpox virus exists today on the planet Earth. Cowpox, another member of the family. Herpes viridae, we've talked about. Herpes simplex virus, both type 1 and type 2. We talked about the herpes virus that causes chickenpox, although I never called it varicella-virus. It is the virus that causes chickenpox, Epstein-Barr virus. No, chickenpox and shingles. Sorry, I'm losing it. Epstein-Barr virus, another herpes virus. Let me shrink that down. Which is actually the virus which causes infectious mononucleosis, as well as a few other diseases. Cytomegalovirus and roseola virus is 2 more members of the herpes viridae. If you don't know about roseola virus, it causes roseola, a disease which 95% of babies get. And it usually happens between 3 months to 9 months of age. So when the babies are very young. All right, any question about any of that? No, let's move on. Some DNA containing animal viruses. The DNA animal viruses generally replicate in the nucleus. Capsid proteins for a DNA animal virus are synthesized in the cytoplasm using host cell enzymes and ribosomes, as well as host cell tRNAs. Once the DNA animal virus enters the host cell, one of 2 things can happen. The first is the viral DNA can be expressed, producing new virions. The viral DNA will be copied. The viral DNA will be transcribed into viral messenger RNA, and that'll be translated into viral proteins, including the capsid proteins, which will come together with the newly replicated viral genome to make the newly made new virions. For multiplication of a DNA animal, oh, wait a minute, let's go back. The second thing that can happen if the virus infects a cell and is a DNA animal virus is that the animal virus can permanently fuse with the host cell's chromosome. This is not called a prophage because it's not a bacteriophage virus. It's an animal virus. It's called a provirus, where the animal DNA virus inserts itself into a host chromosome, the cell's host chromosome. That fusion of the animal DNA virus to the host DNA is permanent. It will be permanently there for as long as that cell survives, meaning the viral DNA will never be excised from that host cell's DNA. And then if the virus is inside the host chromosome, whenever the host replicates its DNA, it will replicate the viral genome. And that will replicate the virus. It'll have 2 copies of its genome now, and both of them inside the host cell's DNA. But there will be 2 cells which have that animal DNA virus. Any questions about any of that? All right, I've talked about that one. So let's talk about multiplication of a DNA virus in more detail. A DNA animal virus can fuse to its host cell. Once again, the molecules of the virus, called ligands or adhesions, that bind to the host cell, bind its sites on the host cell called receptors. So there are specific molecules on the virus that bind to specific molecules on the host cell. And that's why viruses tend to be very specific with which cell and which species the cell can infect and multiply in. Meaning HIV would not infect the cells of your cat because it lacks the receptor molecules, and HIV cannot grow and replicate in your cat. Any question about any of that? All right. The next stage is penetration, where the virus will be brought into the cell either by endocytosis or by fusion of the cell membrane to the envelope of the virus. The viral DNA will be transcribed into viral messenger RNA. The viral messenger RNA will move into the cytoplasm. And once in the cytoplasm, it will translate the viral messenger RNA into the viral protein. The viral proteins will then self-assemble into viruses. And then the viral genome will be stuffed into the viral protein coat to make the new virions. The virus will then leave the cell either by lysing the cell in the lytic life cycle or by budding out of the cell in the budding life cycle, in which case the virion, the newly replicated virion, will acquire its envelope when it buds through a membrane of the host cell. And those are the 2 main ways a virus can replicate if it's a DNA animal virus. I'm not going to talk about the 3rd way, and that's the way that the persistent viral infection will make a naked virus inside the cell. The virus will be incorporated into a vesicle. The vesicle will then go and fuse with the cell membrane, releasing the virus outside the cell. We're not going to talk about how that virus replicates, but we have talked about the other 2. Any question about any of them? Any question about anything so far? All right, if not, let's just briefly talk about RNA viruses. There are 15 families of RNA viruses shown here. These are animal viruses where their genome is RNA. And there's 15 families of RNA animal viruses that cause disease in human. So these are the names of the families. These are the diseases that the virus family causes. I'm only going to talk about a few of these families. Picornaviridae is the number one family of viruses that cause the human cold. This is also the family of virus that causes hepatitis A and polio, as well as some cases of gastroenteritis. You can know just by all the different diseases that we spoke about that this family is very large and very diverse, more diverse than any other family of virus I know. I don't know if it's the most, but of all the virus families I know, this is the most diverse. Let's see, we'll talk a little bit about Coronaviridae. This is the virus family that causes COVID-19. It is also the second most common family of viruses to cause the human colds, meaning most of human colds, or the number one human colds, is caused by family Picornaviridae. The number two, second most common cause of human colds, is caused by Coronaviridae, the coronaviruses. We'll talk about Retroviridae, Paramyxoviridae. Orthomyxoviridae, I'll just briefly mention, is the family of viruses that cause the flu, human influenza. And then where the heck is it? I'm not seeing it in here. Retroviruses. Oh, there it is there. I missed it. Retroviruses, Retroviridae. These are the viruses that cause retroviral diseases such as HIV and AIDS. And we'll talk a little bit, just a few slides of retroviruses, and then a few slides about Coronaviridae. Any questions about any of that? Any questions about anything at all? If not, I think I'll stop here. This is a good place to stop. For some reason, I got that capitalized. And we have a lot to do. Why didn't that go lowercase? Oh, it's uppercase for the this probably. All right. All right, if there are not any questions, I'll be in the lab from 7 to 7:15 just to answer questions. You do not need to show up in the lab if you do not have questions.
Thank you.
All right.
Thank you.
Just log off and say goodbye. Bye.